Okay. Hi, Emily. Okay,
Hello.
it looks like we have everybody on. Hi, Tara. Hi, Hao.
Hi.
Okay.
Yeah.
For those of you that don't know me, my name is Amber Freed, and I am the executive director of Hao-Fountain, and we have other leadership here as well, Bo Bigelow and Becky Ratz. Our mission is to cure Hao-Fountain syndrome by funding research and identifying more patients. Do we have anybody new on the call and would like to introduce themselves?
I'm new. Hello, everybody. My name is Allison O'Neill. I'm a professor at Wesleyan University. I'm collaborating with Irina Bezanova, and so we've got in my lab uses patient-derived iPSCs to model neurodegenerative diseases. So we have the Hao-Fountain patient iPSCs and their isogenic controls. And so we've been working on looking at how they make neuroprogenitors and how prolific those neuroprogenitors are. And then we've made neurons out of them as well. And we're kind of looking at one, they didn't make great neurons, so that's interesting in itself, but cortical neurons. And then we're also looking at arborization, because that's been shown in other models that maybe USP7 affects how neurons can arborize. So that's what we're studying right now.
So exciting. And I have a slide to discuss next month's virtual lab meeting, which you and Irina will be leading. I always like to remind everybody, check out both podcasts, Stronger Every Day. It's a quick listen, amazing for working out, and I think he really touches on every aspect of the rare life. Becky, did you want to say a couple words about the symposium?
Sure. Signups are open, registration's open to attend our conference, which is going to be September 18th and 19th in Nashville at the Hilton Garden in Brentwood. There's no charge to register. We don't have a registration fee. We have a discounted room rate of $169, and we hope to see everybody there.
It is going to be a great time. And for international people on this call, I highly recommend Nashville. If you're going to go to one city, avoid some of the boring ones and go to Nashville. There are some funding opportunities coming up, uplifting athletes, funds travel scholarships to events like the symposium. And then there are some grants through the American Epilepsy Society, and Hao-Fountain does co-sponsor those grants. Our priorities, we're screening small molecules. We're always looking to further understand the mechanism of disease, establish correlations with larger communities. We are working on a disease concept study and a strategic research plan for this year. As a reminder, we have a full biobank and a resource spreadsheet with everything you could ever ask for. In fact, fibroblasts are being sent around right now, as Hao and Emilia can attest. And we also have new research out from all of the people in this group. We are so excited. Thank you, Emilia and Dr. Berlage and Christian Schaff. Well, just everyone. Irina, this is it's so exciting to open up your email and have the PubMed alert and see USP7 front and center. We are always looking for medical doctors, and it's a really super exciting time right now for rare disease in the US because there is a groundswell and there have been more and more rare disease centers of excellence popping up that actually have a little bit of funding. There's one in Austin that I'm going to meet with next week, and hopefully we're able to establish a critical mass of patients at these centers. Oh, hold on. Our main meeting is going to be Irina and Allison discussing establishing iPSCs as a disease model, and I'm really curious now to hear about the cortical neurons and their lack of growth. Hold on, I'm trying to find Hao's slide. Here we go. We're also going to give 5 minutes to Hao to discuss. Oh, let me stop sharing. This publication and also how to use the tool he established. Hao, I'll let you share your screen and talk about it.
Thank you. Thank you, Amber. Hi, everyone. I'm Hao from Washington University, working in Albert Kames lab and Azabani's lab. Thank you for giving me 5 minutes. We have asked this before. We should have asked this before. But essentially, we have a study that was published earlier this year, which is which is on Cell Reports, that the kind of the cover page that Amber showed. That is the first study to look at USB7 specifically in post-mitotic cells, have a very extensive characterization of the function and also mechanism there. Is of course it's just a small piece of the study over there now, but we think this is a quite systematic analysis, um, not only kind of emphasizing the relevance of the nervous system, but also we have a lot of, as Amber said, tools or a lot of data sets we generated, including we have transcriptomics data with the postmitotic neuron knockout brain. We also have a lot of proteomics data that is actually quite unique for our paper. We collaborated the Steve Gigi's lab at Howard to do very sensitive and multiplex TMT mass spec. And we did quite a few experiments. One of them is IP mass spec of the trap domain of USB7 because we are predominantly looking at substrate of USB7 to understand the molecular function of USB7. And also we have just overall the proteome profiling of both the in vivo cerebral cortex of USB7 knockout mouse, but also we use a acute model to deplete USB7 in primary cortical neurons. And we kind of basically recorded the proteome dynamics over time. And actually also from that effort, we were able to find at least one substrate of USB7, but we do believe that there are probably more hidden the data that waiting for everybody to find out. So just want to kind of put it out there for everybody to use and join the effort to find out the exact mechanism of USB7 and the diseases. Yeah.
Did anybody else have any announcements? Anything they wanted to say before we start? Okay, great. Well, I will hand things over to Dr. Sixma and Galperin to discuss activators.
So this was initiated by Emilia Galperin to have a discussion on sort of regulation of USP7. Do I get that right, Emilia?
Yeah.
And I think the idea was that I would give a bit of an introduction of some things, more generic background of USP7 regulation, and then I think I'll leave it to Emilia to have the discussion. Is that the right way to go? I made a little presentation. Some points, it's a reminder because a lot of this was published over the years. And some is maybe a bit more new. I haven't given a Zoom presentation myself for a while, I realize. Okay, does that show the slides properly? Good. So this is from a review that Robert Kim and I wrote long ago, was papers where we had biochemical evidence of interactions of USP7, or could find papers that Robert could find where the real was biochemical evidence, because all of you know USP7 has been studied for a long time and not everything that's in the literature is correct. So there's many targets, many interactors. I think the new data set from Hao will be very useful and find new interactors specifically in those cell lines. I think that's really, really useful. But the bottom line is there are many interactors. And I guess most people here in this audience know among those interactors and targets, there are a lot of other ubiquitin enzymes. And especially E3 ligases very often crop up with their target. And the most famous example is MDM2, MDM4, the E3 ligase for P53, and P53 itself. And I think Vivian has done beautiful work on that, and others in Toronto showing those interactions directly with the TRAF domain. And either of those can bind, and we don't really understand when the E3 or the target is bound. So this is this is from a grant I didn't get, but this is some of the things we are working on. There are many E3s and targets that USP7 works on, but this is just a little example. We also know some dubs. We found that BRCA1A, for instance, is a dub will bind. There's some data on USP11 in the literature. So there are different dubs and E3 ligase. So it's clear that USP7 itself regulates ubiquitin system, and in that way data can become very complex. Another issue I find is that interaction proteomics is beautiful, and I think by now we have from different efforts in the literature some idea of these possible targets, some, as I mentioned, supported by biochemical evidence, but it doesn't tell the whole story because, for instance, MDM2 we do know is a proper target, P53 is a proper target. They're rarely identified in these interaction proteomics because the affinity is very fast off probably, and so some targets bind very well and others don't. And how this correlates to activity, I think, is something we don't really understand. How does USP7 choose among its targets is, I think, an outstanding question. And sort of complicates, I think, all of our efforts because affinity does not correlate with activity for USP7.
You mind if I ask what you mean by proper target? I work on UB3A, and we don't consider anything a target unless we can show it ubiquitinates in a cell culture system. We see the smear. So are you saying you're doing a similar assay?
I'm not doing anything. This is just what the literature tells me. I'm doing biochemical data because in the cell, if you see that smear, it could still be, because it's interacting with so many E3 ligases, it's actually not so easy to show it's a direct
No, it's not.
effect, because it could be due to you affecting an E3 ligase. So to us as biochemists, the biochemistry would then be needed together with that cell biology. I'm not saying that anything that we would do in vitro alone would tell you.
I'm just trying to figure out what you mean by proper target, right? So I have in the UB3A field, I work with people who are strictly in vitro as well. And so they don't consider anything a target unless they can put the E1, E2, E3, and the ubiquitin ligase together and show it that way. I don't do that, but I do a similar assay where with a GFP tag substrate. And so I'm wondering, when you say proper target, if you mean that that's been done, right? So are you showing that the substrate is deubiquitinated either in vitro or in a cell culture system?
Both, I think. I think I wouldn't consider anything a proper target if we didn't have good evidence for both of those, in vitro and in vivo. But it's not just that. For targets where I think the data are quite good, the affinity of USP7 for that target can still be very different among these targets. And I think MDM2, there's little doubt that I have little doubt that that's a real target, but it doesn't bind very tightly to USP7. So in many interaction proteomic studies, you will not see MDM2, and many other things will crop up. So it was just a comment, not complaint actually, that affinities among the targets vary, and it's something to keep an eye out on. Now, one thing, and I think this is what Emilia wanted me to talk about especially, is that we know USP7 as an enzyme is only active when it still is bound to the catalytic domain. So we showed this initially, and then there was a crystal structure from Genentech where they found how that was bound. It's quite dramatic. This is in vitro kinetic data where you see if you have the catalytic domain alone, the Kcat over Km is 0.5. If you have a target that has a part of p53 in it, it's only a peptide of p53, but it's the part that binds to the TRAF domain, you see that the activity goes up by about 12,000 fold. And that's because now you have the tail and you have the target bound. So both this tail that's bound in the active site and the proper target, in this case, just the peptide, but the p53 peptide really changes the activity very dramatically. And that 12,000 fold in the cell, I think, is important. Which means that any assays that look at activation have to take into account the type of activity that you have on the right target and the full-length protein. And I think that's one of the things Emilia wanted to say. I have two more things to discuss. This is from a paper that Niels Keizer published in Life Science Alliance last year, where we looked at catalytic centers of USPs. For USP7, we know that disaspartate is important, but we found that for other related enzymes, the other aspartate, which is more conserved, this fourth critical residue, is more important. And the idea and thinking about catalytic centers of USPs, there's some careful analysis, and this is more maybe for people also interested in other USPs or trying to extrapolate from other USP data. They don't really necessarily work the same. So for instance, if we use a ubiquitin propargyl, oh, I'm missing an L here, and you look at USP1 and you make the mutants in those third and fourth catalytic residue, you see that the wild type can shift with this ubiquitin. So that means it can cross-link to this propargyl group. If you make a mutant in what's known as one of the catalytic residues, this first aspartate, well, third critical residue, you see you affect that cross-linking a little bit, but not much. Whereas the second aspartate is absolutely crucial for the binding. In USP7, none of these matters enough. We can always get that thing cross-linked. So the catalytic mechanisms of different ubiquitin-specific proteases are not all the same. And USP7 is unique, we find. And the bottom line for Niels's paper was that, for instance, these other USPs would differ in the way they respond to this nucleophilic attack, so this binding cross-linking to the ubiquitin propargyl, and in the importance of the third and fourth residue for a plain ubiquitin rhodamine assay. And then finally, something that was shown by a Japanese group first, I think, and I should have put a reference because I don't know it by head, sorry, where we look at pCNA, chains on pCNA, which is interesting in itself, but not that it's now used mainly as a, in this story, mainly as a model target protein. You can differentiate for DUBs between exo and endo and base cleavage. It was shown before that USP7 in that case cleaves off entire chains. It really prefers to cleave off chains. And we see this for K63 and K48 chains, whereas USP1, that was what we studied, really prefers to do exo cleavage. And this is online in bioRxiv now. This is, if you look at the long chain, so we make chains here, in this case for K63, but we can do the same for K48, see the same pattern. You see USP1, the only thing that's made is a monoubiquitin, and very slowly the chain becomes shorter and shorter, and we actually enrich for the monoubiquitin. Whereas USP7 cleaves off these chains, and you see the chains sort of showing up all the time. So it really likes to cleave off a complete chain rather than cleave off from the end. Good. That was my contribution. So to some of the regulation, the main point I think from Emilia was that we need to think about this fact that it's on the proper target, it acts only if you have the full length. Am I right, Emilia? I'll hand over to you.
I suppose, yeah, that was so I come from the rare disease field and cells and ubiquitin assays and stuff like that. And I will show a few slides in a minute, but my question always was, I mean, in the rare disease field, we always try to understand how the specific variant affect function of the protein. And for some proteins, it's really easy to test because we have enzymatic activity. We have an assay to test this enzymatic activity, and it becomes clear. I'm talking about phosphatases or kinases. It's more straightforward there. But once we sort of encounter protein like USP7, there is no straightforward assay or understanding. At least it wasn't for me. And that's why I sort of a little kind of go on and on. What's going on with, how do we understand what specific mutation does for the function of USP7? And also, will be there differences in the way different variants affect development, morphogenesis, and all other tissue-specific functions? So I can show you a little bit of data that brought me to that question, and it may not be very new data. All right, can you see I guess you see the picture, right? So I come from the Noonan-like rhizopathy field. This is and you can see the picture, right, of the children. Yeah, all right. So this is the disease that caused by mutations in the scaffold proteins that control amplitude of MAP kinase signal, which is the fundamental signaling cascade, does many different things. SHOC2 on its own is, as I mentioned, is a scaffold. It binds various proteins. This is just a closed universe. There are other many more proteins that we know SHOC2 binds in a different manner. And right here we have USP7 And I'm not going to sort of dwell on various functions. I just want to show what we think happens with USP7. And it's kind of an example what Titia just mentioned, how many E3 ligases involved in what USP7 does. We're looking at one substrate. This is the substrate. But what we found over the years that USP7 actually need to deubiquitinate HUE1, which is E3 ligase, and that in a way it controls the function of HUE in this feedback mechanism. HUE then ubiquitinate SHOC2, and altogether it results in reduced amplitude of MAP kinase signals. So of course we thought, all right, that there should be some kind of experiment done to look into how various there should be a crosstalk between MAP kinase pathway and maybe pathogenesis of the Hao-Fountain, and we requested fibroblasts that have a variant, this specific variant. And straightforward experiments for us. Let's look into ubiquitination, and of course these are the fibroblast, also primary fibroblast from a non-affected individual. And these are the fibroblasts that have a mutation. And you can see that ubiquitination levels are much lower overall, presumably or due to some sort of effect of USP7. We don't know exactly, because we don't know exactly how this mutation works or how these variants affect activation of the protein, or activity of the protein, sorry. Nevertheless, we do see that MAP kinase activation is increased, no effect on protein levels, including USP7. We didn't see any changes in USP7 levels. And then we looked into ubiquitination of the SHOC2, the substrate that we're talking about. And of course, we see as well much less ubiquitination on SHOC2, which is indirect USP7, or maybe even direct. We don't know how it works yet, the substrate of USP7. Now, there's another piece of data that I would like to show you. We also found that USP7 is phosphorylated. And I'll skip all these other controls that we did. I'll get into this data. It's again, these are the fibroblasts from a patient. And you can see that USP7 is over-phosphorylated. We think this phosphorylation, we need to gain more data. We think this phosphorylation required for the activity of USP7 in the complex with SHOC2 and HUE. We need more data to support this. We also think it affects binding of USP7 to SHOC2. These are the immunoprecipitation experiments. Normally, this binding is inducible, which means it's sort of very transient. Once pathway is active, USP7 binds to SHOC2, and then it sort of gets off the complex. But in this case, we see more of the transient binding. So that sort of led us to the question, what exactly active USP7 means? What is it doing in our complex? And how do we measure this activity in a sense of what just was discussed in tissues, in vitro, in vivo, in whatever systems? How do we determine this? And are there changes in variants? How all sorts of things are not exactly clear to me. So I actually thought I will go first with these questions, and then Titia will explain it to me how it works. But I think vice versa, it will work as well. So I guess,
sorry,
sure.
Really quick, just to follow up on the previous, on the phosphorylation data. Do you know the phosphorylation site?
No, we don't know. We tried various variants, candidates. We haven't got any good data, and we need to do more robust approach, but it's not experiments we haven't gotten to do successfully yet. Okay. So yeah, that would be a great question. Definitely. So yeah, so for again, if I go by this data, I can tell, my conclusion was that this specific variant is actually more active than the wild type counterpart. But then it comes to like, is that true? How do we assess activity? What do we actually make out of all this data, and how do we sort of go, variants are spread all over through the length of the USP7, some close to the active sites, some in trough domains, some in UBL domains. How do we assess effect of those variants on activity? What are we supposed to do sort of to make it a uniform assay? Is there a uniform assay? Things like that. So I'll stop sharing now. And maybe that's the good point to for the expert on USP7 to chime in.
Well, I don't know if I'm the expert, but I think it's really interesting that you see M225I being active in the cell, because what we see in vitro is that that particular variant is still fairly active, even in the context of a full-length protein. By itself, it's more active than the wild type, just the catalytic domain. But in the context of a full-length protein, which is what you have in the cell, it's, I guess, similar in activity to the wild type. So it's interesting that you see this potentially hyperactivation. But again, just as Titia said, you might be affecting E3 ligase as well, not necessarily directly affecting the shock, right? So maybe it's a HUE that's affected. So it's an interesting.
No, we think it's HUE. We don't actually think it's directly SHOC2. Activity is toward HUE.
Possible. Also, it'd be really interesting to see what a phosphorylation site is. If it affects the substrate binding, if it affects activity, it would be really cool to do it. And I think you can probably do that by NMR too. Since we have all the, we have assignment of the whole protein, we can probably test that. If you can phosphorylate in vitro, we can see the change.
I'd be careful saying that this is an activity change, because you're really not looking at activity there in that assay. And the reason I say that is we've looked at active site mutations in UBE3A, and when we co-transfect those, especially in a specific assay for a particular substrate, what can often happen is you get a decrease in the ubiquitin smear, and it has nothing to do with the activity of the enzyme we put in. It has to do with the sort of protection of the substrate, right? Because it can bind, but it can't ubiquitinate. So it's not actually doing anything to the substrate. It's just getting in the way of the ubiquitin ligase. So you see that decrease in the ubiquitin smear. Just something to think about in terms of deubiquitinases, right? Because this is from the ubiquitin side.
Yeah, and I think, related to that, I think, right, your work and your comments just really highlight the complexity, right? And yes, we can measure activity with purified proteins and ubiquitin rhodamine, and that gives us information. Some of these mutants in the TRAF domain, or where you lose the tail, changing the interactome is going to be a factor. There's just thinking about what are the different data sets that one needs to start to gain confidence that we're gaining traction on a more complete story, a complete story of what's happening. And then when you have all of these different mutations, it can get, complex quickly. And I think, right, obviously we're kind of at the beginning stages of starting to chip away with it with, some biochemistry, with some focused studies. But it's going to be really interesting, I think, right, to evolve over time and understand activity and interactome and how each of those relates to function. There's just so much to still learn.
Yeah, I think here the quantitative mass spec that you did, and also what's his name, the guy in Munich with the inhibitors, those data sets are really, really helpful, I think, to see what's going on in time after you inhibit USP7.
We need to get, more of us and others, more of our ubiquination data sets published, because I think those are just nice to be able to see when you have these nice, clear increase in ubiquination, change in abundance, change in ubiquination, not change in abundance without a change in ubiquination. All the examples, right, exist. But just to start to add more layers, and especially if we can start to do some of these in this particular case in disease relevant samples, as some of that is already happening, right, in the Harper lab or Gigi lab.
There's also a lot of redundancy. I think you touched on that, but I can tell you that, even using cell lines that don't have UB3A, when we do experiments, occasionally our substrate shows a ubiquitance mirror because there are other E3 ligases, right? So this is the same thing for deubiquitinases. You've got to be aware that it's not a one-to-one situation. There's lots of ligases, lots of deubiquitinases. There's, a lot going on in the cell.
Yeah, and I feel like especially, right, as Titia pointed out, with USP7, it seems to just be happy to complex with so many different ligases. And so then it can, proliferate pretty quickly as to how many, substrates can ultimately be touched by the combo of dub plus ligase. And then, as you say, then those ligases might independently have different dubs and
Going back to Emilia's data, I think it's really neat to see a very targeted approach where this is the substrate that's affected, the E3 and the dub, and that you're putting in a disease-causing mutation. Would be really curious to put in some sort of mutation that we know deactivates USP7, or even like removing that C tail, right, which makes it way less active than the mutant you have right now, and see if you have the opposite effect. Right? Do you see that disappearance of that ubiquitination, or do you see more ubiquitination? I think it would be nice to see something complementary.
Sure, I mean, I agree that's kind of, we did try a few other variants, but they all were close to, well, we wanted first, because what we see in, I guess my thing, there's one thing to use patient cells, there's another thing to use cell-based assays. So first thing, we wanted to make sure that we see the same thing in other cells, cultured cells, as we see in fibroblasts. So we tried several variants, and I think, but correct me if I'm wrong, we saw pretty similar things when we use cell versus patient samples. So that was reassuring. So we probably should go what you just suggested and use variants that deactivate. We just don't know what we're getting. When we tried this variant, it was sort of, if you remember, even what do you see in your essays? Does it make sense? Because I sometimes have well, I personally have sometimes trouble to understand how, because of the complexity again of the protein, what do we expect to see? Is that an activating, is it supposed to activate, or is it supposed to deactivate? What is it doing? And how do we decide? That was the beginning, and I guess still a question for me. What's the right way to go about this? I can only see what I see, but how do I interpret this data? That's the question for me.
So if, Irina is right, and this is activating the catalytic domain, but not so much the full length. That would indeed be interesting to see it now without the C-terminal tail, because then it should still activate really, and it should really not need the C-terminal tail maybe. That's more or less the question you are trying to ask, Irina, isn't it?
That would be really interesting to see.
Yeah.
In general,
I always think that tail must be regulated anyway. Sometimes there, sometimes not. And how is that now regulated? When is it there? Who puts it there? Or who helps to put it there? Or when is it not available? Because that's always been a hypothesis that its availability to activate could be regulated.
I'm quite sure it is regulated, and I'm sure that some mutations can cause whether it can now bind or not to the catalytic domain. We need a C223A catalytic mutation. And when we make this mutation, we can see that the C tail now can bind to the catalytic domain better than when it's not there. So obviously some mutations can affect not only the catalytic domain itself, but how this domain can be regulated by the C tail.
Also, this other aspect. So in our case, we deal with scaffold, which I think may as well change the way USP7 interacts with E3 ligases. So we see this bipartite binding where like both domains bind, and I sometimes have difficulties to envision those things. And I'm sure there are other examples of the interacting partners that do sort of the same thing. Those are the adapters that facilitate, right, the folding and then therefore activity and stuff like that. How is this? I mean, the regulatory mechanisms, the changes in binding versus disease variants, all of that need to be somehow explained, I think, and that's sort of adding to the complexity of this whole thing.
Yes, obviously, it's very complex and a lot of things can affect it. But what I think is nice about your data, that you have a very clear effect. And that's sort of been difficult to find a readout like this, for us at least, to find something very clear cut. And in your case, you see a very clear effect going from a lot of ubiquitination to no ubiquitination, which I think is important. To sort of continue and understand why maybe that's the case, but it's a nice readout.
Thank you.
So I guess I should chime in that we've been really hitting hard at Cry1. We've started to get some really nice results where we can use USP7, inactivating drugs to affect Cry1 stability in vivo in a cell line that makes a tagged version of Cry1. We have a couple different tags. So we're in the process of trying to use Arena's constructs. And I would, I'd be interested in anybody's constructs. Any mutations you think are really affecting USP7 function, especially in the context of Cry1, we would like to try in this assay systems because now we've got it working. So we're able to sort of show that we can alter Cry1 stability by altering USP7. So send me anything. I'll test it.
So in that case, with the Cry1, there's a E3 as well, or?
Yeah, there's a SCF complex. And we started getting interested in it mainly because MAGEL2 interacts with USP7, and we've worked on Prader-Willi syndrome and Chauf-Yang syndrome, and we initially were and still are interested in how truncating mutations in MAGEL2 can affect Cry1 stability. So if Cry1 stabilizes USP7, then that is going to increase the stabilization of Cry1, right? If MAGEL2 stabilizes USP7. So if you have mutations in MAGEL2, that will have an indirect effect on USP7, which has a direct effect on Cry1. So we're trying to work those things out in the context of Cry1 over time, right? So Cry1 cycling, which we can look at in our system. So very different from the biochemistry you guys are doing, but like a more functional readout of what happens when Cry1 is not doing what USP7 is not doing what it's supposed to do. And I don't have much data at the moment, but the data that I saw from my student yesterday for, several different experiments with different drugs. And, we haven't gotten to Arena's constructs yet, but we have them in the lab. So we're kind of developing this project now. And we certainly have MAGEL2 truncating mutations we're going to put in there too. So we're trying to sort of tease out the pieces for a grant submission in the summer. That we'll see what happens. I don't know where we'll be submitting that to. But,
I have a question, actually. No, sort of, you mentioned something about very weak affinity of substrates and binding partners, right? So how do we go about this? Like, let's say we now in the lab have different, we have two partners that we can pull in immunoprecipitation relatively easy, but they also transient, or at least two different partners. So how do we go about interpreting in terms of data, in terms of, well, let me back up. We try to put some modeling, alpha folds nowadays are very popular. Everybody wants to do, we know we can biochemically isolate them using antibodies from one hand, from another hand, it's all good. We all come together, but once we try putting them in alpha fold, we cannot find the complex. It does not exist in alpha fold. So, and your sort of comment on weak binding, maybe something to do with that?
Yeah, I think
Yeah.
so. Yeah, I think substrates of dubs are not always predicted by AlphaFold. We haven't tried much with USP7, maybe other people have, to be honest, but with USP1, where PCNA for sure is the target, it will not form a complex.
So what is it just, I remember one time you mentioned something about very sort of nibbing on a substrate, or there was some sort of discussion on how it could, the nature of the binding. Anything not binding fully, just sort of, or sitting there versus leaving. There was some sort of mentioning, I thought.
Off rates, yeah, I think kinetics matters. But I don't think we understand. Other people may have better ideas than me, but I know we are looking at transcription coupled repair. We know UVSSA is bringing in USP7. There's decent data on that. In our in vitro data, it binds much better to another component of this large polymerase associated repair system. But that's never found in mass spec data. Yeah, possibly because there's so much USP7 that the mass spec data go wrong. But anyway, it's very difficult to tease out who's the important interactor, let alone who's the substrate from binding data alone.
So that would suggest that we're missing half of the substrates, partners and such?
Well, this is where this kind of kinetic mass spec data that's, I think, started a paper with Eric Fischer, right? And there's this group in Munich that did a paper on also with inhibitors, very rapid kinetics. You get some ideas. So those data sets are very valuable, I think. Might be interesting for your type of problem as well.
Mm.
Going back to the binding and the alpha fold, I think for AlphaFold, it could be hard if binding is through a disordered motif too. It becomes a much more challenging job for AlphaFold, and a lot of substrates, especially those that bind to trap, are in these disordered motifs, not part of the folded domains. And we were just looking at MAGEL2 recently, and essentially what we see is it's not one single motif that binds. It's essentially an ensemble of motifs, and they all can bind with, some weaker, some stronger. But I think the fact that you have several of them all sort of in close proximity makes this transient dynamic interaction between your substrate and your enzyme. So it may be harder for AlphaFold to pinpoint, but you will actually have a pretty decent affinity just because you have a lot of those weak or not necessarily very strong interactions, but just a lot.
How much study was actually done looking into how adapters affect activity? Because it changes, right? There are several studies published. Adapters change in the fold, right?
Yeah, so right. So I think Vivian published, right, a paper where she can show, and so you also published something about the alpha helix connecting the catalytic domain to the rest of the UBLs. Essentially, the substrate binding probably affects the overall fold of these domains and how they interact with each other, and how close UC tail is now to the catalytic domain can also be affected by binding of partners, such as, whether it's a substrate or whether it's an adaptive protein. I can see how this can affect.
So I've been working on USP7 for almost 25 years, and I'll have to say we now know that viral proteins are much stronger interactors than cellular substrates, at least for all those that I've seemed to have measured over the last X number of years. So we should also figure out what it is about these viral proteins that allows them these much stronger binding. And I haven't figured it out yet. I'd love to one day, but they were all like really, really strong. Whereas cellular substrates, we found a great deal in terms of their interactions. But again, for us, it's always been, we've always used like small peptides to measure our dissociation constant. So that could always have an effect as well. But clearly the viral proteins are always, the viral peptides are always much stronger. So you can always try a peptide binding to your protein that comes from your substrate to see what the binding is.
Yeah. So to really get a substrate binding, I must say we've tried quite a bit of cryo-EM so far, and it remains very floppy. So we'll keep chugging at it, I hope, but it gets harder to convince people to actually put these things on the grid.
So if we have a variant, let's go sort of question back to the disease. We have a variant. What would be the good way to assess the variant? The only activity? Should we look into binding partners, adapters? What's the way to go about this to understand the changes in function of the variant in terms of disease? Not the USP7 itself, but disease.
So I think you're very right. Sorry. You're very right about the fact that the adapters will probably play a very key role. And it might be that in this disease, for example, the mutations that don't seem to involve or affect catalytic activity will be involved in binding the adapter. That would be my best, most educated guess as to what those adapters are, because those adapters are so, GMP synthetase is one of them. It's a really big protein as well, and it can interact with so many parts of USP7, and each adapter is different with different interaction sites. So that might end up being the key. So you might have to find the exact adapter that works with that substrate to see the real effect in cells, would be my guess.
Yeah. That's my feeling as well, is that these different mutants, right? It's some combo effect of changing inherent activity and in many cases changing the interactome. And and presumably, and we might at some point not even be able to just focus on the known interactome. It's entirely possible there are new acquired activities, right? That's getting a little further away from the more basic question of affecting activity, but anything could be happening here with some of these different mutants.
So what do you think the best way to measure that is? I mean, mass spec would be my first guess, but also how do you make the sample? Are we doing IPs? Or are we doing some kind of cross-linking and then seeing if they're stuck together? This idea of affinity has kind of thrown my thinking for a loop because, right, like what if it's an important binding partner, but it's a very low affinity binding partner. So how do you capture that quick?
So I can speak from the ubiquitin ligase side because we found a way to get around this. All these interactions are transient. They're all very rapid. When people tried proteomics, just, you know, whole proteomics for UBE3A mutants, they could not find real substrates. And the reason is because it turns out that maybe 10% of, you know, the substrate is ubiquitinated, right? So, you know, now you've got this other problem of a very low percentage of the substrate interacting and getting ubiquitinated. So what we did was we adapted a method that John Hubritz came up with that allows you to trap the substrate. Now, this is going to work from the UBE3A side, but not so much from the D-ubiquitinase side. And the way it works is you take the ubiquitin and you physically attach it to the ubiquitin ligase, and you put a tag on UBE3A, right? So now when it interacts with the substrate, and there's a couple other nuts and bolts, but when it interacts, it makes a covalent bond to the substrate that's bound to the ubiquitin that's bound to UBE3A, and then you pull the whole thing down and do proteomics, right? When we did that, we got very clean in vivo results in Drosophila. This is a paper that's not out yet, but we've been working on it for years. And every time we test those substrates in a separate system, they turn out to be real UBE3A substrates. Now, how do you do that from the opposite side? How do you do it from the D-ubiquitinase side? Because you're not adding anything, right? We have the advantage that we're adding that ubiquitin. So we presented that ubiquitin in the context of being bound already to UBE3A. So I've been thinking about this problem because I would love to be able to do the same approach, but from the D-ubiquitinase side, and I'm still trying to figure out how that would happen.
I mean, I think it'd be amazing, right? If someone came up with like a slick approach to do that, right? In this case, I think at the moment, you know, in some ways, you know, we can explore known techniques. Turbo ID, bio ID, do these get these transient interactions or not for dubs? You know, we've been doing experiments where we just take the purified protein and use that to look at the interactome. And I can't imagine no one of these is going to be perfect.
But now, you know, you have to realize what we did
Yeah.
was we took that 10% of the ubiquitinated substrate and we enhanced it, right? And then pulled it down and identified it. So trying to do that, it's very needle in a haystack, right? And it's
Yeah.
the same for the D-ubiquitinases. So how do you get through all the needles?
Yeah.
If you keep just throwing needles, all you see is needles. You
Yeah.
know, like you have to find a way to get in there and pull the substrates out. And I'm not saying I have that solution. I'm just presenting the problem in a different way because, you know,
Seems to me that thinking about your approach, Larry, is that it seems to me we got to do some kind of proximity labeling with ubiquitin. Like
people have
all of
tried that
ubiquitin
too.
has some kind of tag, right? And so then,
Yeah.
but then I always worry, as I'm a protein biochemist, even though I do stem cell stuff, and you add some kind of tag to USP7, is it the same or not? Especially if you're looking at these changes with mutations. So
For sure, you cannot put anything on the C terminus because it will never go into the nucleus,
right.
and it will not be active. Both.
But there's been approaches using that in UBE3A research using BioID, I think it's called, right, where you're looking for things that are close to UBE3A and it uses streptavidin, biotin to identify those targets. That's worked. Some of those targets are real. I can say that because I know the guy who did that research. It was Hugo Mayer in Spain, and they've been confirmed. But we found a lot more with this other sort of sucking it out approach, right? And I'm just trying to wrap my head around how you do the opposite because it seems like it's possible. Like maybe it has to do with the ubiquitin itself. You know, if you can find a way to make that ubiquitin so people have tried, you know, using the ubiquitin to pull down the substrate, right? But then it's all relative. you kind of need it to be bound to USP7. You need to find a way that you're linked to USP7 and the substrate's linked now, and you pull both down and you look at everything, right? And I'm not there yet. I don't know how to do that.
I'm throwing out an idea, non-natural amino acids and ubiquitin. Maybe that would, especially that data that's showing that they like to cleave, USP7 prefers to cleave the whole chain off.
Yeah.
You could target that kind of, that linkage and think about some kind of non-native amino acid there. I'm trying to think of some way where it binds to the cleavage and then gets stuck, right? That's what we need. It needs to bind and get stuck.
You need to get it stuck
So,
to USP7. If you can do
yeah,
that, and then that chain is attached to the substrate, now you can identify.
the whole dang deal is stuck together, something like that. Anyway.
I was wondering how people think of the relatively simplistic way to just mutate the catalytic site, one of the catalytic site of either debucanating enzyme or ethyl ligases. I do not see a lot of those strategies in the literature, but I did remember Mikko Rapace's lab once used that for their ligase. I wonder how people think of that as a way to, in a way, trap substrate more in a binding state, or this is more like a case-by-case scenario.
It's all still transient with the UB3A C2A mutation. Like we've never been able to use that as a way to find substrates. And other people have tried, and it just doesn't seem to stick, you know. You need to find a way to covalently link it, right? But it's in and out. It's in and out. Doesn't matter, you know. It's not ubiquitinating, but it's binding transiently.
I guess maybe for ligases, things can be, in a way, simpler. As I saw people, there's a Science Advance paper where people basically split the proximity labeling enzyme. I don't remember they are using BioID or TurboID, but basically put one part of the enzyme to ubiquitin itself, the other part of the proximity labeling enzyme to the ligase, and then they pull down the things when the two parts of the proximity enzyme are like in proximity. I guess bring out the specificity when the substrate get close to the ligases.
I think that's the approach that my friend took. I see.
I was impressed by that paper.
So I guess maybe we should wrap it up because I see some people leaving the meeting. Sort of my initial question is still a question: how do we interpret variants, disease variants, in terms of activity? What does it do to the protein? And I think if we know how it affects the activity, it will be easier to interpret this effect on various tissues that the variant affects, or during morphogenesis or during the maintenance. So I think maybe somehow at some point we can come up with
Here, I'm going to put a paper in the chat that Ugo Mayer published, the guy that I'm telling you about, and it's on deubiquitinating enzymes. So it's actually perfect for this discussion, and it's using BioID. So it's exactly what you just talked about. I'm just going to stick it in there before you guys disappear. Everyone. So that might be of interest. I haven't read it yet.
Would you like to say a few words at the end on activity issue?
No, I'm happy to hear everybody else because, as I said, I only work in vitro, and I think all this work needs to be done in cells as well. I think to test we need full-length protein, but again, sometimes maybe there's regulation of that C-terminal tail. But there's a lot of questions out there that seems clear to me. And what's the best substrate? I think will vary for individual people, but yeah.
As someone new to this area, I just love this question, Emilia, because if we could come up with like, okay, these are the 3 tests you do. I think that the structural stuff, I know doing structural biology is not like quick and easy, but what Irina's doing and like how the tail can interact, I feel like that's a really important piece. How can we figure out like this bulk ubiquitin? That's an easy Western blot to do. Like, okay, is that changing? And, what's some other kind of binding to one of these effector proteins or a ligase? Is there something else that's kind of critical we could measure? Like, I like this, because there's all these mutations. So we have this biobank and we have all the cells in our freezer, but how are we going to assess them? We're still trying to figure that out too.
All right. Well, I suppose it's, I don't want to cut the discussion if somebody, I'm not sure it's my job. If someone wants to add something. Becky, Bo,
So
it's
maybe
your meeting.
to be continued, maybe. It sounds like people have a lot more things to say, and it's a really interesting discussion. So I hate to completely end it here, but maybe we could get back to it at some other point and explore some potential collaborations, like with our foundation, or whatever you need from us, honestly, of course. But thanks everybody for being here today. I appreciate you being here and really excited about this discussion. So thanks everyone. Have a great rest of your day.
Thank you.
Thank you.
Bye-bye.
Bye.